events during the year. So this year's World Immunology Day theme is Immunology Talks to Public Health. And we all know that immunology has become a buzzword after the COVID pandemic. And we today we in the participants, I would like to mention that we have students from Somaya College, around 15 students, and their teacher, again our alumni, Dr. Sharon K. And she is here with her students, and then we have faculty, Dr. Madhu Mohanty, coming from ICMR NIV Mumbai unit, and we have Dr. Vandana coming from ICMR NIIH with her students, and from NIIH we have almost 18 participants here, and then we have our own strength of PhD scholars. And today the event has become very special for NIRCH because we have our two alumni who have really made a mark in their fields joining us and delivering lectures and sharing their knowledge with us. Welcome Rishi and welcome Arun. And we don't address them still formally. So Dr. Rishikesh Pandit and Dr. Arun Pratap Sikar were most welcome.
Thank you.
And so what has immunology spoken to us regarding public health all these years is that immunology is not only relevant for the host defense, but it is also important for therapeutics. It is also important for diagnostics. And that's how we kept a title called Immunotheranostics, Guardians of Public Health. The knowledge of basic immunology has helped us to understand how our immune system defends our body against a wide variety of pathogens. And this has paved the way for innovations in diagnostics and therapeutics. And theranostics is a combination of therapeutics and diagnostics. And this was originally used the term was originally used as a radioactive combination therapy strategy to target metastatic tumors. And immunotheranostics thus, describes the synergy of theranostic procedures in nuclear medicine and immune oncology treatments. And in this view, our event today is drafted as a combination of lectures and quiz to understand the role and importance of immunotheranostics in the field of medicine. And without further delay, let me invite Dr. Shubhada Chiplunkar to give away her welcome address on behalf of Mumbai Immunology Group. Welcome, ma'am.
Thank you. Am I audible, Taruna?
Yes, ma'am. Yes, ma'am.
Yeah. Thank you, Dr. Taruna. It's my pleasure to welcome everyone today to this very special event. We are all celebrating the International Immunology Day, and the background about it has already been given by Dr. Taruna. I just want to mention that this was started by Professor Stefan Kaufmann when he was the president of IUIS. And I was lucky to do my postdoc with Professor Stefan Kaufmann. It's a day which is celebrated now worldwide, and the main idea behind this is to create awareness about immunology, to close that barrier between scientists and the public, and bring immunology to everyone, so that immunology is understood by everyone. Of course, as Taruna mentioned, the COVID pandemic did spread the word of immunity, and this word or was used or misused by everyone throughout. But what we focus on doing, and that's I'm going to more talk about Mumbai Immunology Group, where Taruna is now the secretary of MIG. We are a small group, but we are a registered society, and we started this group in 2010. And activities of MIG, which I'm going to just talk to you about. But these activities we can only take forward because of the wonderful support we receive from the directors of various institutes. And I want to particularly thank Dr. Gitanjali Sachdeva for her encouragement and support in conducting these MIG activities. Only through such support can MIG flourish and take forward what we really want to do. We have been doing training in immunology. We have been conducting lecture series in basics of immunology, and because the MIG EC is represented by very senior scientists from institutes like NIRHCH and NIIH, National Institute of Immunohematology, Radiation Medical Center, also ATRAC, Tata Memorial Center. So we have been able to conduct these lecture series both in basic aspects of immunology and also on very focused topics like in autoimmunity or in reproductive health. Uh, just to mention, during the COVID pandemic, since the focus was on vaccines, there were two very interesting topics which were taken ahead. Dr. Taruna had organized, and it was the seminar at NIRH was on vaccines, protective shields against pathogens, and at the same time at ATRAC, Dr. Jyoti Kode had a very focused theme on inflammation and how to combat inflammation during COVID-19. So these lecture series goes on, and we have done these in colleges like SIES, at RUIA, and some lectures at the Junjunwala College. And we want to expand and continue with these activities, but we do need support and interest from students, from teachers who are engaged in immunology teaching. We also conduct hands-on workshops. And we have not been able to conduct many, but the most interesting ones have been in flow cytometry. And these have been very attended by teachers who are teaching immunology, who are teaching flow cytometry without having seen a flow cytometer. So we had this workshop at ATRAC, and it was a wonderful course, absolute hands-on training. We had another hands-on training in basic techniques in immunology. So they did all cell proliferation, phenotyping, and all assays, functional assays. And so these such workshops can be arranged by the Mumbai Immunology Group. Only we need you all to come forward. And we can join hands and conduct these workshops and lecture series at different colleges and institutes. So this is just in a nutshell what the My Immunology Group has been doing. And of course, Dr. Taruna is on both sides. She is the president of the Indian Immunology Society. She's also the secretary of the Mumbai Immunology Group. So I'm sure she's more keen to carry forward such events. She has already been doing it, but we can do it on a much larger scale. So I want to once again thank the director NIRHCH for hosting this event, Dr. Taruna for selecting the topics, and I want to thank the speakers. I was so interested when I saw the topics, and I'm going to sit through both the lectures today. And I want to thank specifically Dr. Rishikesh Pandit. I went through your paper, very interesting paper, and Dr. Arun Pratap Sikarwar. I welcome both of you. Thank you very much. And to the audience, I want to say that please become members of the Mumbai Immunology Group and spread the word across. Thank you very much. I'm looking forward to the event. Thank you.
Thank you very much, ma'am, for gracing the occasion, although you have been into home shifting, and it's by no means a small thing to shift your home from one city to the other, and still you found time for us. And I must tell the audience that there are some college students who might not be knowing about her. So Professor Shubhada Chiplunkar has been a former director of ACTREC, and she is the leading cancer immunologist of the country. And definitely, she is the nucleus of Mumbai Immunology Group that she has started, initiated, and now she's presiding over it. And we all join hands to thank ma'am once again for this August presence. Thank you so much.
Thank you.
And now we will take you over to Rishikesh, because it is almost like midnight for him. It's 12 in the night for him. And thank you, Rishi, for joining us. And I think you can share your slide.
I'll
Please do that.
Okay, can you see the slides?
Not first your introduction slide. Can you please do that? Okay, I will do it. Yes, that's the thing. Thank you, thank you. So what we did was we made a presentation kind of a thing so that we can apprise and introduce the speakers in a better way. So as you all know, Rishi has been a student at NIRCH, and he did his PhD in biotechnology in 2018 at my department, and he was working in the area of HIV immunology, and he moved on to become a postdoctoral researcher at the vaccine branch National Cancer Institute US, and Dr. Vanav Patel was instrumental in taking him to this PDF lab, because this was earlier a laboratory where Dr. Vanav Patel did his postdoc training. And then now Rishi has moved on to become a research fellow and at the molecular immunology department at NHLBI, NIH US. And he would be talking to us today on expertise in cytokine therapies for cancer and infectious diseases, the latest research he has published and is involved in. And as I mentioned, that his research experience has been in the area of HIV. In fact, he developed an ex vivo assay that mimics vaginal transmission of HIV to test microbicides along with Dr. Raina Ficharova and our department. We joined hands, and Rishi went to her lab as a visitor and then set up this important assay. And immunotherapeutic approaches to reduce the size of HIV, SIV chronic viral reservoir and to achieve a functional cure was the work that he was involved at NCI. And now, of course, he's involved in understanding the in vitro and in vivo effects of gamma chain cytokines. And Rishi has 21 peer-reviewed publications at this young age, and he has had 2 patents with us. And now the field is all yours, Rishi. The dais is all yours. Take over. Thank you.
Can you see my slides? And am I audible?
Yes.
Yeah.
Yes, you are audible, and your slides are there.
Yeah, let me first thank you, ma'am, and thank you, everyone. It's a pleasure to talk to you. When you just ask for a presentation, time doesn't matter. You just ask and I'm there. So it's always a pleasure. And we've been celebrating. I mean, I think we started celebrating the Indian Immunology Day in the institute, and I've been there since the first event that we conducted, and it's always been exciting time with the quiz and with the talk. So it's great. And today we're going to talk about the gamma chain cytokines that I've been working for the last 5 years. Just a quick meme just before I start the presentation. We're going to talk a lot about B cells and T cells today, and they're the ones that actually here that they're there to serve us and protect us from different infections. And the other side of the immune system is, when it just gets off from the radar, there's an autoimmunity, which also we need to address. And there are new therapies that in this field that needs to be addressed and find more solutions towards it. So I'm going to talk about exploring the immunotherapeutic potential of the gamma chain cytokines. Mostly I'm going to talk of 2 of them. So gamma chain cytokines, as we know, IL-2, IL-7, IL-4, IL-9, IL-15, and IL-21, these are the gamma chain cytokines. They all share the common gamma chain, as you can see here in the picture, the ones in the blue, for their signaling. And all these exhibit a broad pleiotropic effects on both the innate and the adaptive arm of the immune system. They are expressed or they are produced by different kinds of immune or epithelial cells. And the receptors, so these cytokines are non-redundant in nature. So they have receptors on different T cell, B cell, different subsets within like the naive cells or the memory cells. And they act via signaling mostly through STATs. And then they are mostly involved in survival, proliferation, and differentiation of immune cells. Currently, I'm working on the aspect of IL-2, but for this presentation, I'm going to talk about IL-15 and IL-7. This work was done at the NCI, the vaccine branch that I was working for the last for the first 4 years of my postdoc. So IL-15 is again a member of a gamma chain family of cytokines. It is a homeostatic cytokine. It's produced by monocytes and dendritic cells, and it basically acts on T cells, the effectors T cells, and the NK cells. So it's important in the maintenance of T cells, and in fact development and maintenance of NK cells. So it also has effects on the CD4, effector CD4 and gamma delta T cells. And in terms of function, it is known to enhance the activation and cytotoxicity by increasing the expression of granzymes and perforins, which are effector molecules for cytotoxic activities on infected cells or cancer cells. And it is also known to enhance antibody-dependent cell cytotoxicity. Um, Early in in early 2000s, and it was thought that the IL-15 is not a single chain molecule as other cytokines are here. It was discovered in the lab, in our lab where I was doing my postdoc, that it is there are two bioactive forms of IL-15 called as the het IL-15, which is the heterodimeric IL-15, which has the single chain and also has the receptor alpha, which is transpresented to the T cells or NK cells. Or there is another form, which is a soluble heterodimeric IL-15, which is also present in plasma. So it doesn't really necessarily have a single chain molecule. It is the bioactive form is a heterodimer. And this natural cytokine can be measured using a specific ELISA, and it is known that it has a longer half-life than a single chain. So earlier, the early research was done with a lot with a single chain, but eventually this is the molecule that has been going forward in terms of clinical trials or its use in therapy. As I mentioned, IL-15 expands CD8 T cells, mostly the effector CD8 T cells and the NK cells, and has been shown to regress tumors in several models. So this was always shown because for having the effects on tumors to regress the tumor size, you need cytotoxic activity, and these cells actually enter the tumors in the presence of IL-15, and that is why it has been explored as an immunotherapeutic agent in several preclinical and clinical models of cancer. And as of today, I can tell you there are over 90 clinical trials with IL-15 and also with the het IL-15 that we have used. And it is a very hot, very interesting molecule. When I joined the lab, we were already working on exploring how IL-15 can be used in different cancer models. So I joined on an ongoing project where we were trying to look at the mechanism of action of IL-15 mediated anti-tumor activity. In this experiment, what we did was we used two cancer models, the MC38 murine colon carcinoma and the TC1 lung epithelial carcinoma models. And what we did is we allowed the tumors to grow in mice, and then we treated the mice with IL-15, giving them 4 sub-Q IP injections of 3 micrograms. And then over a few days, we measured the tumor size. We looked at the phenotype of the tumor infiltrating lymphocytes. We did some functional assays. We did some proteomics and transcriptomics to understand what exactly IL-15 does to the tumors. As you can see here, both the MC38 and the TC1 models, IL-15 led to the gradual, to a decrease in the tumor size, or you can say the gradual progression of the tumors as compared to the controls. So this was expected, but the main idea of this project was to understand what is the mechanism behind this. When we looked at the tumor infiltrating lymphocytes, we saw that the intratumoral CD8 counts were significantly higher in both the models, in the MC38 and the TC1. These CD8s were proliferating as measured by a Ki67, which is a marker for proliferation. We also found that, as a result of an increased CD8 counts, the CD8 is to Treg ratio, which is supposed to be a prognostic marker for, an effective treatment, was also increased, which is a good parameter, a good tick towards looking at the effects of the therapy. And when we looked at the functions of these CD8s, we saw that these CD8s were able to produce interferon gamma. They also were expressing CD107 alpha, which is the degranulation marker, so that they can degranulate and eventually release perforins and cytokines and have their cytotoxic activity going in the tumors. So everything looked good, and we knew what IL-15 would do to the CD8s and in the tumors was doing. So our model was working. We seen similar effects on the NK cells within the tumors. So it was both with CD8s and NKs. One interesting molecule that we found was when we did the transcriptomics of the tumor infiltrating lymphocytes. Of course, you would expect granzymes to come up and perforins because these are part of the CD8s and NK cells, which are already higher in the tumors. But we also saw something called as XCL1, which is a chemokine that attracts XCR1 cells. And very, very little is known about the XCL1. And this was one of the top hits that we found in the tumors after IL-15 treatment. So we measured, actually, we got we actually had an in-house ELISA that we created for this, and we measured the XCL levels in the tumors, and indeed the levels were higher after in the IL-15 treated group. And XCL typically is produced by NKs and CD8 T cells. And we were able to actually see that after IL-15 treatment, the NKs and the CD8s were expressing more NK. So we knew the source of XCL1 in the tumors after IL-15 treatment. So we then started asking, what is the implication of XCL1 induction within the tumors? And what are the XCR1 positive cells? Because they are the main cells that are going to come in response to the XCL1. When we also when we were analyzing the transcriptomics data in depth, we also found that DCs were enriched, the dendritic cells, the population of dendritic cells were enriched in the IL-15 group, along with the cytotoxic or CD8 T cells. And the conventional dendritic cell 1, which are actually the most potent in production of interferon-dependent chemokines and functions, do have the XCR1. They do express the XCR1 positive. So this is a gating strategy of how we really looked at or found the CDC1 cells. They are characterized by they're of course lineage negative. They express MHC class 2. They are CD11C positive, and from there we look at CD103. And they express the transcription factor IRF8. When we got these cells, we were happy to see that in response to IL-15 and the increased XCL1 by the CD8s and NKs, we could see that these cells, the CDC1 cells, were significantly upregulated in the IL-15 treated both cancer models. So what we knew was IL-15 was able to bring in a myeloid cell population in the tumors as a result of the XCL1 that was secreted by CD8s and NKs. So as I mentioned, these cells are potent in production of the interferon-dependent CXCL9 and CXCL10. These are chemokines that eventually attract more effector or tumor-infiltrating cytotoxic cells within the tumor. And what we saw was they were actually able to produce CXCL9 in response to IL-15 within the tumors. And we also measured the intratumor levels of these chemokines, the CXCL9 and CXCL10, and they were they were significantly higher in the IL-15 treated group. The result of this is that the higher expression of CXCL9 and 10 would attract cells that express the receptor CXCR3. And these cells are typically an effector CD8 T cells and NK cells, which are able to kill the tumor cells. So we came up with a mechanism of action where treatment IP injections of IL-15 induces XCL1 and also promotes NK and CD8 proliferation within the tumors. This XCL1 brings in the XCR1 positive CDC1s, which in response to interferon gamma starts producing a lot of CXCL9 and CXCL10, which again brings in the CXCR3 positive effector cells which eventually leads to the regression of tumor sizes. So this was the mechanism that we found. But one interesting conclusion was that the systemic delivery of cytokines like IL-15 leads to a multi-pronged effect, and the effects goes beyond the direct targets, which would just be CD8s or NKs. It goes beyond, and it's the collaboration of the myeloid and the lymphoid cells that together result in the robust tumor regression. So you just can't look at one single aspect of, okay, look at the target cells. It definitely has many more effects, and all of that eventually add up to the functional efficacy or of the immunotherapy. And after this, while I was doing this, we wanted to start a new project on IL-7, and unlike that, I jumped on an ongoing IL-15 project. I was just given IL-7, and I was told that, look, we need to know how to use IL-7, and so it was a new project that I began, and it was easy because it was another member of the gamma chain cytokine family, although it was completely different than IL-15. IL-7 typically, the IL-7 receptor is present on naive and memory cells. It hardly has any effects on effector cells. It's more important in maintaining memory and naive cells. And I'm going to now show you some data that we recently published in iScience, where we show that the treatment of IL-7 actually just alters the immune cell landscape in blood and lymph nodes. And this was very exciting because there were many new aspects that we actually stumbled upon as we were exploring this molecule. Just to give you a brief introduction of what IL-7 is. So IL-7 is a critical cytokine in both the development and functions. So it has peripheral roles, and it has also important roles in the thymic development of T cells. It's a critical growth factor and survival factor for T cells. And it is an indispensable molecule for replenishing the T cell pool. And because of these functions, IL-7 therapy is in the clinic for the last 15 years. It's typically been used to treat conditions like lymphopenia, which is typically seen in bone marrow transplantation cases, patients undergoing hemodialysis, chemotherapy. It has been used extensively in chronic HIV infection, where the CD4 counts are significantly lower. Sepsis associated with lymphopenia, and IL-7 is doing wonders in the sepsis patients. Also in severe COVID-19 patients where lymphopenia is associated, IL-7 therapy is considered to be working and has potential there. Obviously, IL-7 has also been used as monotherapy or combination therapy in several immunotherapeutic therapy protocols for cancer, in combination with anti-PD-1, in combination with CTLA-4 antibody. One thing is all these studies, preclinical, clinical, all studies have definitely showed that IL-7 is very safe and expands the T cell pool, a function that is expected and known. There are two companies, biotech companies, Revimmune and the NeoImmunotech, that are working full-fledged on developing IL-7 in different clinical settings and different conditions. So it's a very lucrative, very hot molecule in the gamma chain cytokines, along with IL-2 and IL-15. What we did in this study was we used a non-human primate model to understand how IL-7 can be used. And there are many benefits of using that. Of course, for the cancer model, we don't have a cancer non-human primate model. But apart from that, this model system is very close to humans. You can do a lot of long-term longitudinal studies. You can understand the pharmacokinetics and dynamics in detail that can be done. And these are excellent model systems for HIV, SIV infections, which also I was working extensively on. So we had these to explore. And for cytokine therapy, it's very critical that you have the right understanding of how you optimize the dose. You eventually want to give the minimum dose and have maximum effects. And for that, you have to just tweak on how you treat, when you treat, and how do you get best effects out of it, how you optimize the use of cytokine. Also, there are a lot of side effects associated with free cytokine. So you have to make sure that there is the cytokine is bound to the receptor and is doing this function immediately, because there are a lot of side effects associated with free, which could be just inflammation at the site of injection to anaphylaxis. It could go in that array. And you eventually want a sustained growth. You don't want a transient growth. You want a sustained growth and survival of the responding cells. So you need to have a legit dose optimization done before you understand how these cytokines can be used. What we did is we implied a novel dose escalation strategy. We actually, to begin with, we made a rhesus macaque specific IL-7 because we were treating rhesus macaques. And we used a novel dose escalation strategy where we gave 6 monkeys 50, 100, and 200 microgram per kg sub-Q injections of IL-7. These injections were given 3 days apart. And 2 days after the last injection, the animals were necropsied because we wanted to understand systemic effects. And we did a lot of transcriptomics, understanding phenotypes. The idea of dose escalation was when you have some cells that have the receptors for instance, IL-7 receptor, and there is very free cytokine in the plasma. When you give a small dose of the cytokine, very small dose, um, you generate a small pool, a gradual pool of the responding cells, which have now more receptors, which can take the incoming increasing dose, which would be the second dose in this case. This way, all the all the second dose will be occupied by the new cells that are generated, and then they're ready to have another big dose. This way, you optimize the use. There is very free cytokine that can be measured, and this avoids side effects. So we've used this protocol for even IL-15 studies in monkeys, and it is way superior than using a standard fixed dose regime. To start with, we started understanding the pharmacokinetics. And as you can see here, after giving IL-7 every 4 hours, there was a rise. There was a significant peak in the levels of IL-7 in the plasma in these monkeys. And these peaks were proportional to the dose that we were giving. So 50 had a smaller peak, 200 had the highest peak. The levels of IL-7 could be measured 48 hours even after the IL-7 after IL-7 injections. So it had a longer time that it was in the plasma, so it could do its effect. It was largely very safe because we did a biochemistry and we saw all the parameters apart from a few enzymes that were transiently upregulated. We saw that all other parameters were within limits. So we could say that this dose regime was safe in monkeys. And then we looked at the effects, the pharmacodynamics. We looked at the effects on the cells that it is supposed to respond. So just to give you an understanding of who produces the IL-7 receptor, because these are the responding cells, it's typically the central memory and the naive cells, as you can see here, that they produce, express this CD127, which is the IL-7 receptor. And as the cell transits from the central memory to an effector memory, it downregulates CD127. So these are the least responsive in the as compared to the rest of the two populations. And when we looked at the CD4s and the CD8 T cells in the blood, in the PBMCs, and we looked at the markers for proliferation and survival after IL-7 treatment, both in the central memory, in the effector memory as well, and the naives, they were significantly up. And they were also surviving better as measured by BCL2. So as you can see, the naives were surviving better. The central memories were proliferating better. And this is how IL-7 treatment differed between the two, even if the receptor expression was similar. Then we looked at whether this effect was systemic. After a sub-Q injection, typically the highest effect is seen in the draining lymph node. So it is important to see that these effects are gone beyond the draining lymph node. So we saw that like lymph nodes collected from different anatomical sites like axillary lymph node, inguinal lymph node, mesenterics, mediastinal, spleen, which is also considered as a secondary lymphoid organ. When we looked at the proliferation of, I'm sorry about that, with the proliferation of CD4s and CD8s, we saw that they were significantly up as compared to the control animals. Again, in the lymph nodes, when we looked at Ki67 and BCL2, as we did in PBMCs, they were up in the treated animals after treatment with IL-7. Just to give you a sense of what are lymph nodes and what they do. So lymph nodes are secondary lymphoid organs. They are the place where a primary adaptive immune responses are generated, and they are at different anatomical sites. And if you look at the lymph node, they have a very well structured T cell area and a B cell follicle. In the T cell area, the knives the knives become memory cells with an exposure with an antigen, and in the B cell follicle, the antibody response is generated. And the movement of B and T cells in the lymph node is triggered by an array of chemokine receptors. For instance, the chemokine CCL19 and CCL21 are produced in the T cell zone of the lymph nodes, and they bring in the CCR7 positive T cells, which are resident in the T cell area. On the other hand, the CXCL13, which is a chemokine that is specific for CXCR5, the receptor is CXCR5, brings in the B cells and the T follicular helper cells in the B cell area. So it is a tight a network of these chemokines and chemokine receptors that actually are responsible for homing of these lymphocytes in specific areas. When we looked at levels of chemokines and cytokines and growth factors in IL-7 treated monkeys in the plasma, what we saw is several chemokines and growth factors and cytokines were upregulated after IL-7, and it was also a dose-dependent effect. Interesting was the CXCL13 and the CCL19 that I just talked about. Important homing of T and B cells in the lymph node. And when we did a transcriptomic analysis of the lymph node cells after IL-7 treatment, we saw that the transcripts of CXCL13 and CCL19 were significantly upregulated in the lymph nodes, suggesting that these chemokine sources in the plasma were from the lymph nodes. So obviously the question was, does this affect the entry of CXCR5 positive, typically B cells, or the CCR7 positive cells inside the lymph nodes, like the central memory cells? So in search of that, when we looked at what happens when you treat monkeys with IL-7 just 4 hours after the injection, what we saw is the central memory population, which is CD25 high and 28 high, typically, was significantly lower in both CD4s and CD8s immediately 4 hours after the injection. You can see it from 37 to 22, from 24 to 13. And these are the cells, the central memory, that express high levels of CCR7, the chemokine, receptor for CCL19, which is produced in the lymph nodes after IL-7 treatment. And when we looked at the frequency of these cells in the lymph nodes, they were significantly upregulated. So suggesting that in response to the CCL19 that is produced by IL-7, these cells were able to home in the lymph nodes. Similarly, in response to the CXCL13 in the lymph nodes, we would expect the B cells to home, which have the receptor CXCR5. And indeed it was. What we saw in that after IL-7 treatment, the B cell frequency in the blood was lower, while the T cell proportionally went up, changing the ratio of how the B is to T was in the blood. And there was a dramatic increase in the B cell ratio in the lymph nodes. So the B cells moved from the periphery into the lymph nodes, resulting into an inversion of a B is to T cell ratio in the lymph nodes. Typically, the T cells dominated the lymph node before IL-7 treatment. Now it was the B cells. And this was remarkable because IL-7 receptor is not expressed by B cells. So this was a complete indirect effect as a result of the CXCL13 gradient. We also looked at what kind of B cells, and we more characterized this finding. And we saw that there were certainly more follicles. If you can look at the red arrows, there were more red arrows after the IL-7 treatment. CD20 is the marker for B cell. And these B cells were germinal center. Germinal center B cells are actively proliferating. And as you can see, there was just one lit for BCL6, which was BCL6 is a specific marker for germinal center activity. And they were significantly upregulated, or there were a lot more germinal centers after IL-7 treatment. So this was pretty striking. So we looked at the other arm of the B cell, which is the TFH, and we wanted to see what is the effect on TFH. So just to tell you the basic function, in the B cell follicle, the TFH helps the B cell, and this interaction helps in the germinal center formation, affinity maturation, class switching. It produces IL-21, another gamma chain cytokine, which is important in the whole B cell maturation process. And interestingly, as a T cell progresses from a CXCR5 positive to a GC, which is germinal center rich TFH, as it progresses with an expression of PD-1, it starts down regulating the IL-7 receptor. So the least of them to respond to IL-7 would be the germinal center TFH, because they, as you can see here, they express the least amount of CD127, which is the IL-7 receptor. However, what we found was after IL-7 treatment, there was a marked increase in the proportion of these cells, the germinal center TFH, and not so much, and not the other type of T cells. And as a result, and this was as a result of the CXCL13 gradient, because they were not IL-7 responsive directly. When we also looked at imaging, we saw that these cells had high BCL6, which is again a germinal center specific marker, and you can see here that, and they were proliferating. So these CD4 cells in the follicle were significantly higher. We also saw that after IL-7 treatment, there was an increased expression of IL-21. As I mentioned, it is a marker for activated TFH. It is produced by activated TFH cells, so it was significantly higher. So what we saw was IL-7 treatment dramatically increased the entire follicular activity, with increasing the number of B cells, with having more follicular germinal center formations, and then more TFH. And the most intriguing part was this effect is on the cells that lack the IL-7 receptor. So these were a completely indirect effects as a result of chemokines that were because that were actually induced by lymph node cells as a result of IL-7, systemic IL-7 treatment. We also came across two genes that were very interesting, FLT3 ligand and IRF7, which were significantly increased that we saw in transcriptomics. And FLT3 ligand is actually binds to the FLT3, which is the CD135 receptor, present mostly on the multipotent progenitor and common lymphoid progenitor cells. And it has been shown that high levels of FLT3 in mice leads to a dramatic increase in the number of plasmacytoid and conventional dendritic cells. And specifically, the plasmacytoid dendritic cells are the ones that express IRF7. This is a transcription factor and a major regulator of the interferon alpha gene. And the PDCs, or the plasmacytoid dendritic cells, are the major producers of interferon alpha in our body. They are the first line of innate immune, first line defense cells. So we were wondering, okay, so then do we have any effect on the plasmacytoid dendritic cells after IL-7 treatment? Typically these cells are lineage negative. They express MHC class 2, and they express CD123, the receptor, and CD4, and this is how you identify PDCs. And when we identified that, we saw a significant increase in their frequency in the blood. It was almost with a mean of 1.5-fold increase in the blood. And these cells which were in the blood were expressing increased CCR7 expression, which made us believe that as the T cells moved from periphery to the lymph node in response to the CCL19, these PDCs would also migrate into the lymph nodes. And when we looked at the lymph nodes, there was a dramatic effect, almost a 10-fold increase in the PDCs in the lymph nodes after IL-7 treatment, which was almost, they were very little, and it was a significant increase in all the animals. And this increase was also seen in the spleens as when we compared to control animals. So there was a higher frequency of PDCs. When we looked at the functionality of these PDCs, in response to TLR7 and TLR9 ligands, typically they produce interferon alpha. And as a result of increased PDCs in the lymph nodes, we also saw increased interferon alpha production in the lymph nodes. This was also associated with an increased interferon alpha gene signature in the lymph nodes. As you can see, all the genes marked in blue are a typical interferon alpha related signature, which was seen after the IL-7 treatment in the lymph nodes. So taken together, we in this new dose escalation protocol, we obviously established what was known, that it systemically promotes proliferation and survival of CD4 and CD8 T cells, but we went much beyond just this basic finding, which is a common report of all preclinical and clinical trials. We went ahead to conclude that IL-7 treatment accumulates the central memory cells in response to the CCL19. There is a massive infiltration of B cell in response to CXCL13. This results into not just an accumulation of B cells, but increased in B cell activity, as shown by germinal center formation and TFH activity. And on the other side, on the myeloid arm of the immune system, we saw that IL-7 increased the frequency of PDCs, which are able to produce interferon alpha and upregulates an interferon alpha gene signature within the lymph nodes. So this was pretty striking. And going forward, you could just imagine, as a result of an increased B cell activity and follicle activity going forward, it just makes a wonderful case that IL-7 can be used as a vaccine enhancer to improve antibody responses in so many conditions, especially with HIV and HIV vaccines, where getting neutralizing potent antibodies has been a challenge. So this is our way to move forward and see whether we find something. Just to have some final conclusions, I'm just going to make a very general comment on what lessons I have learned from cytokine immunotherapy. Of course, it has a wide range of applications, including cancer and infectious diseases, now autoimmunity. It does what it's supposed to do. It systemically expands the cells that express the receptor, and there is a lot of scope that we need to still explore about how to optimize the dose, how to limit the side effects. And we should not miss out the multi-pronged effects that we see in both the cases we saw in the case of IL-15 and in the case of IL-7, that it goes beyond the direct effects. We see a lot of chemokine releases from specific tissues. In case of IL-15, I showed you in the tumors. In IL-7, it was in the lymph nodes. That results into specific cell trafficking. Also, there was an induction of a lot of other cytokines and growth factors that results into an increased frequency of other cell types, like the myeloid cells that I showed, the PDCs. And there is a complex interaction of different cell types that together bring an effective immunotherapy. Of course, these molecules are also used in ex vivo expansion of adoptive cell transfers, the CAR T generations to maintain stemness. And there's a lot to explore in terms of how these can be used to get a very effective CAR T therapy or generation. With this, I would like to thank all my lab members who helped me from the NCI, who helped me take these projects forward. And I just want to plug in at the end of the presentation that I'm co-editing a topic on frontiers where we are we are reviewing different projects and papers regarding gamma chain cytokine research. And please, you're welcome to contribute if you have anything in this field. So thank you so much, and I'm open to take questions.
Thank you very much, Rishi. It was an amazing talk and very informative. And I'm really proud of you that you've made a mark in the field of immunology. And definitely, I'm sure students have loads of questions. And in the interest of time, we'll take a few for you. Yes, I knew Dr. Vaina was going to ask you something. So, Dr. Vaina, can I request you to come here on the dais? Yeah, because then it'll be, since the recording is going on, so it'll be easier.
So, hi Rishi,
Hello.
and thank you. It was both nostalgic and refreshing.
I'm
So,
sure.
I just want to first say that it felt wonderful to see that 2008 JBC paper that you cited. It's 15 years since that work began in that lab. I was on that paper, and it just shows, when you start good work and you continue doing good work on it, it never gets old, and it keeps getting relevant. So that is the first thing. Thank you. The second thing is, I'll ask you one question for the IL-15 and one for the 7. So for the 15, from what I recall, the earlier work used to deliver IL-15 using DNA plasmids,
Correct. Correct.
right? And considering that the beta gamma receptor is a kind of a, let's say, it's a receptor that's used by other cytokines too, right? So it's basically the high affinity alpha receptor that confers specificity of the cytokine IL-15 compared to, let's say, IL-2. Now, in your studies, you used protein, if I'm not
Correct.
mistaken, right?
Yeah.
And you used relatively higher doses of protein. So what I'm interested to know is, were you not interested in figuring out what low doses would do? Because the IL-15 receptor complex, as has been shown, is a highly stable complex. And I thought the original work was all about saying that you didn't need to use very high doses and you could achieve bioavailability and activity with low doses, even that delivered by plasmids. So I'd be interested to know why you decided to go for using protein, which is in high quantity, which could have acted on cells which may not have had the CD25 but would have had beta gamma, right? So if you can just comment on that first.
I mean, the honest answer is we went below 3. We went tenfold below 3 or whatever, and the effects that we started seeing were 3 and above, 3, 6, 10. So we went to the minimum dose that we could see effects. But I could say if I give 1 microgram, I would hardly see any infiltrating CD8 T in response to in the tumors. And actually, even like now I work with IL-2, and having the explanation that you gave, IL-2 typically now in my hands or in my experience, anything below 30 micrograms as compared to an IL-15 3, it's completely comparable. So you need way more cytokines than, and that is why I think the idea of giving a DNA-based IL-15 did not fly eventually, is because we needed way more levels of the cytokine to see the effects.
Okay, so thanks. And with respect to IL-7, the one question I have is, you've shown obviously in a kind of a de novo administration that the IL-7 can do things, right? It can induce
Yeah.
enhancement of memory and potentially enhance all this. So, we've known about this in HIV for some time, about using IL-7 in clinical trials. But one major problem, if I've understood the literature correct, is that IL-7 has not been able to repair damaged lymphoid architecture. Can you correct me, or do you know if that is still the case? Because see, it's okay, just one sec. If it's okay to give it prophylactically, it's fine. As a vaccine inducer, that's great. But majority of PLHIV are now infected and have undergone gut damage and microbial translocation and all of that. So anything relating to repair or restoration of tissue architecture?
Yeah, so certainly not. I mean, that is one of the reasons in 2020, IL-7 in chronic HIV infections stopped. The clinical trials were stopped. And what eventually the big conclusion was actually it was expanding the pool of infected cells as well, because these are central memory cells. So eventually
Correct.
you were expanding the pool of infected cells.
And the reservoir, basically.
The reservoir, which is latent. So that made it so basically the idea when we started IL-7 was not to use in HIV. It was certainly to find how it can be used, but we were convinced that it is just not a good candidate in the chronic HIV infection.
Thanks, Ruchi. Thanks a lot. And all the best.
Thank you. Bye-bye.
Dr. Chiplumkar, would you like to ask something to Rishi? Yeah.
First, I want to congratulate him for a very nice piece of work and a very lucid presentation.
Thank you.
I have two questions. See, basically, these what you explained, they are chemoattractant. They work through the mechanism of. I'm talking in terms of tumor infiltration. Okay, they're working in terms of like chemoattractants, bringing in a lot of lymphocytes and cell types to the tumor microenvironment. How do they overcome finally exhaustion?
They do express PD. I mean, in case of IL-15, we did an extensive understanding of what the PD-1 expression was, and they do express PD-1, high PD-1s. So they bring in exhausted cells as well, because they are the ones that express CXCR3, like the ligands that they come in response to. So I think going forward, the combination therapy is what now people are trying, anti-PD-1 with IL-15.
Right.
So that is more potent, or even radiation analysis.
Did you look at other exhaustion markers like TIM3, lag, and others, on these?
Yeah, we did.
Like, do they make a difference in the expression of exhaustion markers?
No, the expression of PD-1 and TIGIT specifically, I can say, were significantly up, whereas TIM3, lag3, were up. So the CD8s that brought in were high PD-1 as well. That was for sure.
So they may not be functionally,
but
they may not be doing the function.
But when we saw their response to produce interferon gamma, or to have, at least when we got the cells out and we re-stimulated them to see interferon, they did produce very well, and they were PD-1 positive, because we could phenotype which are the cells that are producing interferon. So PD-1 positive were producing interferon gamma. So now in the tumor environment, we don't know. This is ex vivo, when you bring it out. But I think to overcome that switch, we need a combination therapy with anti-PD-1. That is going on. I mean, that is a clinical trial that's already ongoing. And I'm sure it'll show better.
And my second comment more is like, this appears like a traffic jam situation. right? And
Yeah.
when we are talking either of CAR-Ts or of any other form of, how do you regulate it, because this will be an overload.
Yeah, so these are definitely high levels, especially IL-5, IL-7. If you go to see, we are giving 50, 100, and when we were giving 50, 100, we thought we are giving too much. And then, we saw now papers are, people are giving 1,000 microgram per kg, and this is in clinic. This is in clinic with the companies doing, and so these effects, the cytokines are safe for sure, but now as you mentioned, the pleiotropic effects, I think we still don't know. I mean, our study at least came up with some, and we were trying to look at how beneficial it could be, but I'm sure there are going to be effects that we need to overcome of these excessive cytokine treatment.
And last one now. And since you
Yeah,
mentioned about IL-2, you're working on IL-2.
yeah.
Capillary leak syndrome has been a major problem with IL-2. And that is starting from the days when Lopez started using it. And
Yeah.
so how does one overcome this? I was just
So
toying on the idea of, is there anything like metronomic therapy, giving in very, very small doses over a
yeah,
long
so
period? Because
IL-2
that
a
has been a
small.
serious problem, the capillary
Yeah,
leak
yeah.
syndrome.
Yeah, so IL-2 definitely, toxicity of an IL-2 is an issue since 80s. I mean, they've from the time they've started. Low doses induces Tregs, which is not good for the cancer immunotherapy, because that is something that you don't want. Um, the thing that I'm going to just say, what I'm working on, we are making designer molecules. We are changing the interaction of its interaction with beta gamma chains, and we are trying to make either a super IL-2, so the very low dose, which is, we call it as H9 background. So, these are very low dose doing very high activity as compared to, because we fine tune how it regulates phosphostat 5 activity. Uh, and then the other way is we go the other side of the spectrum. We attenuate completely so that we can use it specifically to just improve Tregs, for instance, in case of autoimmunity, and has no effect on other CD8. So we are exploring those kind of designer IL-2 molecules currently. So
Thank you once again for a wonderful talk.
thank you so much.
So, Rishikesh, good to see you and wonderful, nice work. So, I just had one query regarding IL-15. Uh, since Werner was also here, he already asked you about IL-15. So, we've been speaking about IL-15 and its role in immunotherapy. And in fact, when we were working on COVID, SARS-CoV-2 infection, we also saw that there have been reports where it is also proposed to be one of the immunotherapy candidates. So, however, when we conducted our study, we saw a little bit something of a contrasting effect. What we saw was there was an increased association of IL-15 levels with severity, in fact, increased hospital stay, and also a somewhat negative correlation with frequencies of all other immune cells like lymphopenia,
Yeah.
particularly. So, what are your comments on this? Like, how are these contrasting? One thing could be, like, I can help. Sorry, I'm preempting. It
Mhm.
could be a compensatory response, increase in IL-15. But if one has to really use it for immunotherapy, how would we balance these effects?
Yeah, so I mean, as it's exactly said, lymphopenia. When you see lymphopenia, you will see IL-15 upregulation. These are homeostatic cytokines. They will come up in response to the lower A receptors when they sense that. So that is certainly there. And IL-15 immunotherapy. I mean, in when I was looking at IL-15 in chronic in severe COVID patients, I think it would. I mean, there people have shown that it increases the T cell counts, but the toxicity or the other effects of IL-15 treatment over an IL-7 treatment, IL-7 would be a better candidate than IL-15, certainly because the kind of uh, other cytokines and chemokines that it brings, like TNF alpha or IL-6 expression with IL-15 is way systemic delivery of IL-15 is way higher, which definitely is going to add to the severity. So definitely, I think in terms of severe COVID patients, I think IL-7 is the way that people would go than IL-15. but it's tried.
Okay.
I'm sure both are tried.
No, yeah, I guess maybe it's not such a straightforward
No, I mean, people
response.
have tried
We
IL-2
really
as
need
well.
to, yeah, because yeah, maybe it does have a role, but we need to probably understand a little bit more.
Yeah.
Yeah, thank you, Rishi.
So, Rishi, time to formally thank you for a very interesting talk and also the QA session. You can see how much interest your talk has generated. And I'm sure there was a time when we used to understand that, cytokines go up and down all the time. So we really can't make any meaning out of them, neither for diagnosis nor for therapy, because they're pleiotropic. They have so many effects, each cytokine. And so we don't know how specifically we can use them for therapies. But we have really moved forward, and your studies in macaques and in the cancer models have definitely shown promise. And we wish you all the best.
Thank you.
To do very well in future, and we'll keep calling you.
Yeah.
Thank you.
Thank you. Thank you.
So now we switch gears and we move on to another speaker of ours, another alumni of NIRCH, Dr. Arun Pratap Sikarwar. And he has so much of knowledge, not limited to biology, physics, astronomy, planets, and so much knowledge that he was the first person I interacted with when I came to NIRCH in 2007, and we have been together since then. We have been in contact, and he's an avid reader, reads. I must say he's not reading, he's actually absorbing like a sponge from various books. And I encourage all our participants to imbibe some of his knowledge that he's going to share today with you. And an important introduction about the topic that he has chosen is that we all understand that for every nation, resources are important. And human resource is the most important resource. And for human resource to act or to contribute, you need a healthy human resource. And a healthy human resource is directly linked to the public health. Any pandemics can seriously hamper your human resource that we have seen during COVID times. And that's how Arun is going to tell you more about the health and economics behind it. And he is at this moment assistant professor at the Dayalbagh Educational Institute, Agra. He's an educator and scientist of cognitive science, neuropsychology, and brain biology. He did PhD in biochemistry in 2008 at the Department of Molecular Immunology and Microbiology with Professor KVR Reddy and at our institute. And he has been a research associate at JNCSR Bangalore, and then he did diploma in Indian patent law and practice at Bangalore, and then postdoctoral associate he was at Aarhus University, Denmark, and then postdoctoral associate in neurosciences in 2014 at Weill Cornell Medicine, Qatar, and he did MBA in education administration in 2014 from Symbiosis, Pune. He's a nominated speaker for TEDx Talks and a resource person to UGC HRDC Center at JNU, secretary of Kalam Center, invited by various media houses. He's on TV many times for live sessions, telecasts, and debates on mental health and stress. And not to delay further, over to you, Arun.
Thank you so much. Thank you, madam. I'm so happy that I'm joining to NIRCH, right, where I did my PhD. Okay, so my slides are visible to you? It's slides? Okay.
Yes, your slides are visible. Yeah.
Okay, so So after Rishi's presentation over, very basics of immunology and, bottom of approach of, IL, interleukins and other, basic immunology, I will be doing my presentation over, top-down approach, probably because I'm a science communicator. And I took the topic on a holistic way, like how a country should be ready in the near future whenever such kind of pandemics happens, unfortunately again. So the topic, vaccination induced herd or hybrid immunity, a potent armor against pandemics and bioterrorism. Let me have my association with this, occasion, immunology. That time it was not molecular immunology and microbiology when I was in NIR H. So this used to be immunology lab. Okay, just one second. It used to be the immunology lab. So I have my association, even though my work was not totally over immunology. And I'm sure some of you would be attending this presentation right now. Although Dr. Reddy sir is now superannuated. So after having my remembrance of NIRCH and immunology lab, so every day in the world, there are some, anniversary, birthday, or any, memorable days. Just yesterday, on 24th of April, it used to be the World Malaria Day. And after a few days, on 29th April, it shall be World Immunology Day. And Dr. Chiplunka ma'am has already told about Stephen Sachi Kaufman's contribution as a International Day of Immunology. Okay. And we are after 3 years of COVID-19 disease that has, catapulted this, whole humanity over the globe so much. So this 29th, although we are running just 4 days before to that. Now, so this year's theme of Day of Immunology is Immunology Talks to Public Health. So being public and science communicator over televisions or through newspaper or magazines, I want to take very holistic and easy and supple topic of this immunology of COVID-19 and how a country should face in upcoming problems. So as you all know that in a medical college, there are more than 12 or more than 14 departments, and they are like digest. So even animal physiology or human physiology have digestive system, cardiovascular system, integumentary. Similarly, there's nervous system where right now I'm having my more time goes on nervous system teaching and studying. So it is the master organ, and immune system is for the defense system, like how a person can survive the whole life expectancy as much as possible without getting any disease or unfortunate death. So these two systems are very important for nervous system that controls your body, coordinate your messages, and make you to live as long as possible, and immune system to keep you safe. So during the COVID-19 pandemic from March 2022 till today, like almost 3 years have been passed, and we had a very tough time of fear and awe of COVID-19 infection that has done a lot of casualties, lot of, pessimism. Not only biologists, but even non-biologists, like I can say Bada Pav wala, like I can remember the Mumbai with Bada Pav or Chai wala, even people who have deprived of formal education. I don't say illiterate because many and people who have not formally educated, but they are even more literate than the degree holders. I would say they also know the meanings of immunology nowadays, as Professor Chiplankar also explained. After COVID-19, immunology is a now buzzword. So immunology has its origin in the study of how the body protects itself against infectious diseases caused by 5 kinds of microorganisms, and these are viruses, bacteria, protozoa, fungi, and helminths and worms. After these, 2 are most notorious, viruses and bacteria related to the human health. Okay, so so believe it or not, at least every day we we, unknowingly take up to 1 million microorganisms, including fungi, to our body. Okay. It means there's something inside our body that keep us safe. It means we are every time under the influence of viruses, bacteria, fungi, and much more in the environment. And this load of pathogens or bacteria, viruses, or microorganism, I can say, depends on the environment, such as humidity, rainfall, solar radiation, and carbon dioxide level in the environment. And you can think of having flu when there's a rain or when the season is changing, like summer to winter or so, how we get infected with the cold and flu and other diseases. So I take to my students of UG and PG how this immune system is similar to like an army or soldiers. So immune cells are the soldiers that Rishikesh has explained very nicely just now. So immune system of the body is like a defense system or army of police that sentinel each and every second or moment to keep our body safe. Until like yesterday evening, when I was on the COVID-19 World Health Organization site, up to 0.763 billion people have, confirmed COVID-19 in last 3 years or so. It means 1 upon 10. I can say like up to 7.5 billion people, 0.76 have confirmed COVID-19 interaction, and almost 7 million or 70 lakh people have been died so far, as per the government records by this COVID-19. Now, during the COVID-19 pandemic, there were a lot of, misinformation, disinformation, and malinformation. Over some television, I was, explaining this, when the COVID-19 was full-fledged, that there's a misinformation in the environment that inadvertently people used to tell and keep on sending here to there, just like WhatsApp university messages. But disinformation is deliberately spread, and malinformation during the COVID-19 was specifically to harm. Okay. So these three things were, leading, a lot of, time consumption of public in the COVID-19 that led to the lot of stress and anxiety, even suicides, even family conflicts, and great loss of education because education institutes were closed. It but led to the onset of yoga and meditation throughout the world. Although in 2015 International Yoga Day started getting celebrated on 21st of June, but during the COVID-19, because it's a respiratory disease, so pranayama was very much famous. And it also led to good news for Ayurveda, that is phyto compounds or phytomedicines used during the COVID-19 that were immunoregulatory or immunoprotectant, I can say, and a lot of Kada and concoctions, these were in the market that led to the onset and revival of Indian civilization of knowledge or traditional knowledge of phyto compounds during the COVID-19. So the rationale behind my topic is COVID-19 the last pandemic? I mean, is it the last, and now we are so mature and human civilization is having so much control over medical science that there will be no pandemic in the future, or there will be no endemic or pandemic? And my answer is no. It is not the last pandemic, and it is just start probably. There will be certainly the medical science is not yet matured enough, even though US and UK and even some other countries are doing very well, and they're coming up with a lot of immunotherapeutics, just like you were telling, this case was telling about new compounds are coming up, but still it is not that sufficient. How to keep human survival on priority in the world, having disruptive technologies and ever-evolving geopolitics of hate and animosity of greedy countries to show off their superiority. Although I can say there are a lot of, United Nations, G20 and Quad and several, treaties of different countries, but the bitter reality lies that many countries are polarized now, especially after Ukraine and Russia conflict, and probably who knows that next conflict will be in Southeast Asia. It could be somewhere near Taiwan or Indian Ocean. We don't know. And and we are also having disruptive technologies day by day. Like Richard Moore's principle says that every 2 or 3 years, technology becomes double and size becomes half. So there are a lot of changes are happening, and nowadays there are no conventional war of, like I can say, army and air force. It is through the satellite or drones. Right now drones are working very well in such conflict. Now there's need of herd immunity and hybrid immunity to be achieved as early as possible to safeguard people whenever there's an unfortunate onset of pandemic or endemic or forced endemic or pandemic. Even there's a lot of stories behind that COVID-19 was man-made, okay? And there are a lot of theories behind that, a lot of proofs have been coming up. So now let me take you the difference between endemic, epidemic, and pandemic. Endemic is like localized in the foci here and there. Epidemics, very severe, but more than endemic, but restrained to one geophysical location over the globe, and pandemic throughout the world. So pandemics is when epidemics occurs worldwide, and COVID-19 was one of them. And we all are fortunate that we have come across last 3 years to know about this pandemic. And this COVID-19 pandemic was the sixth pandemic after flu of 1920, Spanish flu. So this spectrum of microorganisms. So these are 5 microorganisms very notorious related to the medical science: bacteria, viruses, protozoa, fungus, and helminths. So it leads to the infectious diseases and disorders. Many organisms live in or on our body, like ectoparasites remain in our body, however many are inside. However, I'm not saying that all the microorganisms are notorious to a human body or medical science. There's gut microbiome because it comes under neurosciences, and gut feelings also, the term comes up, that there's a healthy microbiome is very necessary for our brain and mind and our thinking and our mood. Okay, so I'm not saying, I'm not blaming all the microbiome is notorious and detrimental to medical biology perspective. But under certain conditions, some microorganisms may cause diseases. Some infectious disease may be passed from one person to other person. And there are a lot of stories of zoonotic diseases also. Some are transmitted by insects or other animals. We may get others by consuming contaminated food or water. And in my opinion and in my research, maybe the next pandemic could be food or water associated. Or it could, other than this present ongoing airborne disease of COVID-19. This infographic shows so far, like so many pandemics have happened. In 21st century, there were 3: Ebola, MERS, that is Middle East Respiratory Syndrome, and severe acute respiratory syndrome of in the 21st century. Then in 20th century, it used to be the Spanish flu that led to up to 50 million deaths. And before that also in the history, like 1520 smallpox, then plague of Justinian led to, onset of, like in 5th century, another way. Remember, our COVID-19, this present ongoing COVID-19 has led up to 7 million deaths. So it is sizable amount. And this is the 6th pandemic after Spanish flu. And there are great chances that every year, statistically up to like 2% chances are there that another new disease or pandemic may come up. So this way, if a person is living like 70 years of age, so the chances statistically that up to 50% more chances one can get interaction of get such pandemic. So communicable diseases, infectious and transmissible diseases are illness that result from the infection, presence, and growth of pathogenic or capable of causing disease biological agents in an individual. And some diseases which are under not control of human mankind is neglected tropical and vector-borne disease. Tuberculosis is bacterial disease, obviously. Coronavirus, viral, dengue virus, hepatitis, climate change and health, measles, HIV, AIDS, all this, So a number of communicable diseases can be significant health threat to the local, regional, and global level, lead to the epidemics and pandemics. Now, sovereignty of a country is very much important, and that leads to the variety of like almost aggregation of some 200 countries over the globe. Okay. And the concept lays out basic rules for how countries are allowed to interact with one another. In principle, it means countries get to control what happens inside their border and can't interfere in what happens elsewhere. This shows the sovereignty. This is the also hallmark of United Nations. But but it is not so, or in reality, in practical. Many countries keep interfering other countries, and that is the bitter truth. Because entropy is part of the physics, and so the world will also be, having arm races and financial gains by some clever countries. So although sovereignty is hallmark, but in practical it is not so. There are also greed of expansionism and super powerfulness by some countries, especially in Asia. Also, some countries are very notorious and known for this expansionism and to show and flex of super powerful nature. So COVID-19 pandemic is one of the biggest challenges that humans have faced in 21st century. It has been a painful reminder that no one is safe until everyone is safe. You remember, it's very important sentence. No one is safe until everyone is safe. COVID-19 has demonstrated in stark terms that a pandemic is a complex phenomenon fueled by a multitude of diverse, interconnected factors and revealed deeply rooted structural weaknesses and insufficiencies in our health, socio-economic, environmental, and political system. And you might remember how India was helping the countries, especially African countries and other countries, by giving Covaxin and Covishield, those vaccines. Although there was school of thought running that why not we should safeguard our public first, but government think of this empathy and they were helping other countries simultaneously. So present pandemic is an opportunity for better preparedness for the next pandemic, if any. So it's true. Uh, so being better prepared for the next infectious threat requires a mindset shift and the adoption of approaches different to what we have used this time. Foresight can support the transition from emergency response to proactive infectious threat planning and management. Right now, because of COVID, up to 2% GDP has been spent by government of India. And we are expecting that within few years or so, up to 3% will be spent over health support in the country because just last week or so, India has come up with like 145 crore as the number one country in the human population. Now, how to control such pandemic? So first way is prevention is better than the reaction, or prevention is better than the cure. Global action is needed. Real-time monitoring of any unknown disease is necessary. Freely and honest sharing of medical data should also be adopted by all the countries, and that will be done by the World Health Organization and other verticals of United Nations, that they should share data honestly. However, it was not honest in COVID-19, that everyone knows. They should share, they should tell the to know about the history of or onset of such disease. Microbes are key part of the wildlife diversity and often drive population dynamics that already I've explained. But the greedy nature of human being is affecting the land degradation and habitat loss. Now, habitat loss and land degradation is very much affecting the microorganisms in the nature. Recently, since last 1 month, and I was over one television just few days back, all of a sudden, a dog's bite has become so severe that dogs are biting to death. I mean, several human beings. Even, excuse me. So genetic spillover is also one of the problem. And now COVID-19 was famous because of bat. Bat was having effect of genetic spillover, and that led to the COVID-19. So there are up to 1.7 million undiscovered viruses, and up to 40% of them can infect mammals and birds, including human beings. So there are quite chances that after 1.7 million, several of them can become fatal to mankind and be pathogenic. So how to combat this unfortunate sudden onset of an epidemic or pandemic? So by transmission, we can stop the transmission, or we can use the treatment or by giving therapeutic drugs, or we can safeguard the public, remaining public, whomsoever has not got the disease or death, by giving vaccination. So protein, now vaccination is given by several ways, like by protein subunit, inactivated virus, or some other ways. So how many years it takes to for a drug to become available for any use? Like recently when COVID-19 was just initiated, it takes like there was no therapeutic drug available because it takes up to 10 to 15 years, uh, including all the 3 phases of clinical trials. It's a lengthy procedure that everyone knows. And 1 out of 10,000 chemical compounds from the chemistry hardly reaches up to the all the three or four phases of clinical trials and become a commercially available drug. And many of this, in a triangle, uh, gets, succumbed off during this period. So in the COVID-19 pandemic, ivermectin was the first, drug repurposing drug that doctors used to give in a hope that it was uh, giving positive result for getting getting treated from COVID-19 infection. Similarly, in the second and third, phase of COVID-19, remdesivir injection was quite, famous. And there was a lot of black marketing of remdesivir injection, and people were spending up to 1 lakh, even though it was drug repurposed. And there was no such report that it was very much, COVID-19 specific molecule. But even people were using because we were helpless and we were not having any solution. So COVID-19 has, made this vaccinology a separate science, and now governments are considering vaccinology is important aspect. So like Covaxin was inactivated virus, yes, and Covishield was, replication uh, replication deficient, a vaccine for COVID-19. Similarly, mRNA, DNA-based vaccines also came up in the market. However, Pfizer story is very unfortunate and shocking that this uh, genomic vaccine was not so successful as it was claimed for. So SARS-CoV-2 vaccine needs like immunogenicity. It's that compound should give uh, exciting the immune system in the human body. And so spike proteins were, the main, target by which this antibody was made in several ways. These, other than bacteria and viruses, like fungi are also one of the challenging thing for pandemics or endemics, it being forced upon by some clever country. But penfungal vaccine is incredibly challenging to formulate, and no successful vaccine available in the market so far. Maybe because of fungal cells are eukaryotic cells and humans are also eukaryotic. And probably this is beyond my expertise in the immunology, that how so far vaccines are not available for fungal pathogens like Candida neoformans or Candida or Aspergillus fumigatus. But there is a quite danger that this disease or fungal diseases can be used as a bioterrorism or related to any unconventional war in the future. So modes of onset of epidemics and pandemics. So the modes could be air, water, food, vector-borne like mosquito, or exchange of bodily fluid. Right now, this COVID-19 is associated with the air, and next threat could be through the water-borne disease also, okay? We do not know, but it could be a possibility. And vector-borne diseases like malaria and elephantiasis, etc., we have already come across in our textbooks. So next threat could be through the waterborne pandemic. Now you remember there's in Asia there's a Himalaya mountain range, and several northern India rivers, they originate from the Tibet. And you already know that who owns Tibet right now, even though it was an independent country before 1950. How and why it is being, taken over by some country. And why, especially northern India, Pakistan also is having, Bhutan, Bangladesh, I like several rivers coming from this side, from north to south, come from there. In India, southern India is having their own rivers, so probably they are having less danger. But in my opinion, northern India, having especially Gangetic, Yamuna, and Satluj, these rivers are having, uh, can have an impact in, and can have an importance in this uh, epidemic or pandemic in near future or so, because it is originate. Air we need every second, and water we need every 1 or 2 hours, or I can say few hours. So remember, water is also very much necessary for survival of human being. Food one can omit for days or so. Food can be changed from here to there, but there's no substitute of water and air, remember. So these are two important aspects related to any pandemic or epidemic in near future. So this idiom comes very much, frequently, all is fair in love and war. So like 50 years or before, there used to be 1-to-1 fight, or soldiers to soldier, and they fight one by one. Nowadays you can see a lot of missiles getting fired, or drones going and taking over as a payload those bombs. And in near future there could be war on the space by affecting the satellites, by affecting the transmission, by affecting the communication between a country. That could be possibility. So India would have now probably third army very soon, other than Navy, Air Force, and Army, to as a space army. And who knows, in near future also there will be mind crisis, or like are you minding your mind, or you minding, or is it minding you? I mean, people will hijack the mind, okay? Like Neuralink project, you might have heard of, that can be negatively used to affect the mind. So there will be no free will in near future. It could be in 100 years or so. So that time the war will be uh, war will be having worst by this way also. Now, coming to many infectious diseases such as measles and chickenpox can be prevented by vaccines. Frequent and thorough hand washing and practicing hygiene help us from most of the infectious disease, and that is the hallmark of Swachh Bharat Abhiyan also, that is leading to cleanliness drive by the government of India, and it is giving positive results throughout the country. If there is a cleanliness in the environment, so the viral and bacterial load becomes minimum or permissible, or threshold level. It will reduce down, so there will be less chances of at least airborne or waterborne diseases. So this thing is going on correctly at the right time. There was one article showing emerging trends of edible vaccine therapy for combating human disease, especially COVID-19, pros and cons. I mean, this topic related several other articles I have studied in just last few days, where researchers are doing efforts to develop an effective, reliable, and easily available vaccine candidate to protect against COVID-19 and any other future pandemic or endemics, like if there's any health situation comes up. And in last 3 years or so, more than 200 vaccines have been worked upon. Several have been a successful story. Several are in the market. And indeed, these vaccines have saved the humanity, I can say. So even though therapeutic drug is still not, available so far, but vaccines have led to the herd immunity, and that led to our survival. Plants are also considered as a potential source for the production of monoclonal antibodies, vaccines, drugs, and immunomodulatory proteins, as well as a bioreactor of factories for the bulk protein production. Nowadays, because plants are associated with the mankind since antiquity, and plants are very much part of our amino acids, our building blocks, or our food. So why not this immune system or vaccinology can be incorporated through the plants for the mankind? So the characteristic of a vaccine are like several other factors it comply with. The scientific evidence is enlightened that plants are the rich source of oral vaccine, which can be given either by eating the edible parts of the plants or by oral administration of highly refined proteins. The use of plant-based edible vaccine is an emerging trend as it possesses minimum or no side effect compared with the synthetic vaccines. So imagine a situation that by the use of genetic engineering, by the use of plant biotechnology, by the use of vaccinology, we can get immunization of a disease through these plant products. That could be a fruit, that could be salad, that could be vegetable or so. Now, edible vaccine will be better option because these conventional vaccines are needed with a cold chain. It also need a viniculture and vaccine and syringes, and syringes also need to be very much new every time, so it leads to the plastic wastage or so. So I just forecast that why not it will be easy and quick delivery of vaccine through the plant-based. Although this research is in infancy and people are coming up. Recently to the Niti Aayog, the health section of Niti Aayog, I have sent one proposal that why not Niti Aayog should ask the ICR and other bodies that they must start asking the proposals for edible vaccines. So let's have a one opportunity that can be used as a quick vaccination or taking herd immunity along with conventional vaccines of uh, these intramuscular or other potent candidates of vaccinations. So now it needs genetic engineering and biotechnology knowledge, plant biology knowledge, and vaccinology. And one can use, desired gene leading to the production of the protein. That could be, suppose in case of a virus, it is um, capsid protein for the COVID-19, then transgenic crops, and then it could be in the form of tomato, cucumber, carrot, lettuce. I mean, that is part and parcel of our day-to-day food habits. And then it can give a very quick round of, vaccination and herd immunity. Now, recently, just last month, Government of India and Prime Minister Office has come up with this uh, inception of INTSAPI, that is India Coalition of Epidemic Preparedness Innovations. India to invest in developing any vaccine within 100 days. And Prime Minister Office has made sure that within 100 days, would it be possible for a scientific community, especially the scientists of you people, ICMR and DBT, that we can come up with the solution of a vaccine formation within 100 days. So even government is going to take help of ICAR and Director General of Medical Research, and how to make generics and vaccines on a large scale. And this is very much urgent need of today's for the Government of India to safeguard our 145 crore of people in future. Okay, and government is very serious for that. And now, also in my one of discussions with the Government of India, secretary, I told about there's a need of real-time water pathogen check alongside water pollution in the northern India rivers. So I gave my hypothesis that there should be real-time check, and nowadays there's no need of human-based laboratories. There could be Internet of Things, IoT-based remote sensing technologies or probes inserted here and there in the northern India, especially in those rivers, because the water is coming from Tibet and China. And we do not know if accidentally or if there's any spillover happens or so, or intentionally if something bad happens, at least we can avert and avoid any waterborne pandemics in near future. And there's no need even for mankind to be there checking. Even data can be taken by the internet and cloud, by Internet of Things. Now, another important work right now is coming IOMT. So this umbrella term of IoT changed and beyond to IOMT, that is Internet of Medical Things. Now, Internet of Medical Things is a collection of medical devices and applications that connect to healthcare information technology systems through online computer network. And nowadays, you might have now almost all of us, even I'm having my watch, smartwatch, having a health app, and this data goes to the internet. And this data can be given to a physician. It can be shared with the elder people. It could be, your father, mother, or family member, or caretaker. And this way, real-time health monitoring is under the control and under the strict vigilance. Okay. And nowadays, even this actigraphy watches are having the data of are hormonal. Like, it can tell you the stress, it can anxiety. It could be, in a prognostic way. It can also relate to early diagnosis of a disease, or like if there's any probable disease. Recently, Strand Biotechnology has been taken over by Reliance, and they're going to set up like $145 uh, exome sequencing of human beings, and this exome sequencing will be used for uh, ancestry generation of mankind, and also to real-time health, or sorry, to health predictions. So I can presume that IOMT, along with like just like US is having 23andMe, so India will have probably this very soon by the Reliance, related to this saliva. One is to give only saliva, and the saliva will give the DNA, and then exome sequencing, and then giving the lot of data of the health perspective and predictions of future. So this all can be incorporated together, can be used for real-time check of health. Now, just last month or so, Ava app has come up in our mobile phones. It is Ayushman Bharat Health Account, Ava. Even my phone is having. Now, this Ava will have all the data of a person's medical history, or medical examinations, or pathology reports. And this Ava app will be synced with the hospital. So if a person is moving like Delhi to Chennai, or Tirunelveli, anywhere, so a person is not needed having carrying the medical data. Rather, by Ava app, one can, like, sooner or later it will be joined with Aadhaar card also. So this way one can have a check on human health in real time for all the people. Now, there's quite danger, and there's a second school of thought saying that why a person should disclose the medical data that could be used for bad purpose. But every time these disruptive technologies come at the cost of some disadvantage. Even though like online payment nowadays is having very good implication, like 19% people are working happily with this, but there's quite little chances of bank forgery. So probably this real-time data monitoring will be maybe somebody will use for any notorious purpose also. So the take-home message from my lecture, and because it was not related to very basics of immunology, it was more on top-down approach. So there's a dynamic global geopolitics will move from conventional wars of army, navy, and air force to non-conventional war strategies such as bioweapons in near future. It is inevitable truth. I can't say, but it's bitter truth. COVID-19 was airborne pandemic, and who knows, next could be waterborne pandemic also. There's urgent need of check systems of river, especially in northern India, to keep a vigil over any suspected pathogen or disease-causing microorganism. Just like pollution control is being monitored nowadays, so other than pollution, we can check the pathogen load also. Internal security of a nation would need not only soldiers, but medical researcher and health scientists. And I still remember ICMRs, like NIRCH was having this COVID-19 detection. Remember, even in Agra, Jalma Institute of ICMR was having a COVID-19 detection facility in the very early phase of this COVID-19 by real-time PCR. So medical researcher and health scientists will also be called like as a health soldier, I can say. There should be another term. They are not less than a soldier. Surveillance system of human disease should be on priority. Vaccinology lab should be set up throughout the country, at least in the north, east, west, and all the directions, and especially in the major cities here and there. use of online platforms until necessary be adopted to avoid person-to-person contacting. I mean, just like I'm having my presentation in online mode, rather avoiding to go to Mumbai because it would have taken 1 or 2 days, can be done like that, at least in today's world. And use of holistic medications, including Ayurveda and yoga and meditation, should be the part of our day-to-day life. That will also lead to the betterment of our immune system and better fighting capability of any disease. And it is proven fact that Ayurveda, yoga, and meditation have beneficial effect, and that has given very positive results in the COVID-19 pandemic. And our country was not that worst, like unlike other countries. Meditation affect through the pituitary gland to you know hypothalamus to pituitary to all endocrine glands, and there are several reports that meditation affect our thinking and mood and our mind, and that mind also lead to the positive effect of uh, counteracting any disease or getting rid of disease or getting free of disease very quickly. Because in COVID-19 many people have story that where those people were very strong enough mind wise, they couldn't get disease and they were happy. So with this poem of alma mater and my respect to my alma mater NIRCH, I would like to say thank you to invite me, and I'm happy that I could join NIRCS today. Thank you so much. I can take now questions.
Yes, yes. Thank you, Arun. And an amazing talk and a very big message that water could be the next source of infection, a pandemic. And I understand from Dr. Jayashankar's, one of the uh, I think he was addressing some civil services people, and he was telling that India by 2047 is going to be the one of the top 3 economies. We could be the first one as well. That's what he said. So when you are doing so well economically, you know that we will have more enemies, and we have to be more careful because once upon a time India was it's working. Yeah. So once upon a time, India was called Sone ki Chidiya, and then we have seen those days when we were, not even independent to exercise, our will, free will, and now we have again, taken a leap, and we are going to the top of the world economy, and we are, our decisions and our opinions are really mattering to the world. So what I will conclude is that your lecture has given us more questions, more food for thought, and we need to be more alert from our health point of view, from our public health point of view. And definitely immunology gives us that knowledge base using which we can go for edible vaccines, as you said, and our government is being readying for preparing a vaccine and delivering a vaccine within 100 days. And I think we have made a mark on the public health on the global scene, and we'll continue to do so. Thank you.
Oh, yes. So the economic prosperity of country will lead, a lot of animosity and people, enemy becoming enemy of the country, and that will lead to the any unconventional, a kind of, conflict, because conventional wartime is now over. They will do all this clandestine job, and that is my worry. And I have written one article in one this army research development magazine also, where I proclaim that this kind of thing may happen. I'm very pessimistic, but it should not be so. But who knows? It could be so also. Yes.
Yeah. Thank you, Arun. And may I request if anyone has any query? Dr. Clara is sitting in the audience, Arun.
Okay. Yeah.
Yeah. Gauri ma'am is there.
Oh,
Dr.
yes.
Clara is there. Yeah. So your lab colleagues are listening to you.
Yeah, I
And
shared one
proud of you.
photo. I shared one photo, and I was thinking that few of them will be there to listen me
Yes.
and might be seeing me in a different avatar, because that time I was just a kid for them.
Yeah. And would you like to say something, Dr. Clara? Okay. I think in the interest of time, everyone is saying now we should start our quiz contest. And thank you very much, Arun.
Thank you.
And it was a pleasure to be a host for you. And we look forward to many more such talks from you. Thank you.
Thank you. Thank you.
Thank you, Dr. Chiplunkar, for being with us for so long. And we'll now take over to we'll give the dais to the students of
But
innate
before that,
immunity
I just want to thank
Yes.
Dr. Arun for because his talk was really keeping with the theme of bringing immunology talks to public health. Thank you very much. A very provocative and a very nice talk. Thank you.
Thank you, ma'am. Thank you so much.
Grateful. Miss Hajra and Miss Rutvija, please take over for the quiz contest.
Okay, I take leave, Sharuna. Thank you very much.
Sure.
A wonderful event.
I can also take a leave, ma'am. Thank you.
Yes. Thank you, Arun. Thank you.